Restarting. Okay, perfect. Okay, and I am going to share my screen in just a second here. We'll start off with chapter 15. Share screen. Okay, so chapter 15. So I have the topics to review, our little study guide next to me here. So it starts off with review the structural and genetic differences and similarities between X and Y chromosomes. So let's go ahead and find that information in our, let's go straight to our pictures here. All right, so this is an excellent place to start. So in this picture off to the side, we see how there's an X chromosome and a Y chromosome. The chromosomes themselves, they don't look like an X and a Y, right? They're just a linear molecule. We could have called them molecule A and B, right? We just say X and Y. That's just what scientists chose to call them. But the difference between these, they are sex chromosomes. So they contain the genes within them that are responsible for individuals developing a uterus versus developing testicles, right? Uh, the Y chromosome is much smaller than the X. And typically, the Y chromosome contains our SRY, or some people call it the SRY gene. And this gene, once expressed, it produces the proteins necessary to trigger the development of testes. We can see how these are not homologous chromosomes, right? Remember, homologous chromosomes, they're like pairs of socks. They are the same shape, same size, and code for the same traits. So an X and a Y, they're not homologous. However, it's still important that during meiosis that they do end up aligning next to each other at the proper time so that they can be segregated or sorted correctly into the gametes. So if they're so different, what allows them to align to one another during meiosis? Well, it has to do with the little bit of homology that they do share on their tips. All right, so there is 6% homology between the X and Y chromosome. There's a little bit on both tips, and that there will allow them to align properly during anaphase of so that they align properly and then separate properly during anaphase of meiosis 1. All right, so that's very important. So even though they're very different, this 6% homology does allow them to pair and align. Let's see. Any questions about that?
No.
Okay, cool. All right, so next on the list, it says review the differences between XX and XY individuals. And I just put a little note in there. Just remember when I use the term male and female, I'm typically describing like someone that's a typical sperm producer versus a typical egg producer, just to keep it simple. So the differences between XX and XY, the main difference I want us all to focus on is that, especially when it comes to X-linked traits, an individual that's XY, it does not have a backup X. So if there is a mutant allele on their X, then they have the affected trait that we're following. So they are affected with that trait for sure. However, if a female or a typical female, right, egg producer that is XX, they have a backup X. They have two X's. So they may have a mutant allele on one X and then a normal wild type allele that's dominant on the other. And as long as they have one of the normal dominant alleles, then that trait that we're following, maybe it's color blindness, that trait will not affect that individual because they do have one copy of the allele that is functioning properly in order to allow them to see color. In order for an XX individual to be colorblind, they would have to have both of their X's having that mutant allele on it. Right. And remember that XY individuals, they donate their Y to their sons and their X's to their daughters, right? That always happens. So a son from somebody that is XY never has the X from the father, always has the Y from the father. And sons of a couple will always have that son their X will always come from their mother, not their father, because they got the, Y from their father. Okay. All right. Let's go on and look at some pedigree charts. That's the next on the list here, and see some of those patterns of inheritance. I have 3 that I am going to focus on. I believe that's all we did in here, and I got to find them. So let me go through real quick. So the first one talks about X-linked recessive traits. Oh, here it is. There we are. And it's color blindness is our example, which is good. Let me see if I can make my screen. There it goes. So let's see if I can do this. You don't need to see all my messy notes. X-linked recessive mutations in humans, color blindness. We see how this pedigree chart, we know that this is an X-linked recessive mutation or trait that we're following. Why? Well, because I notice how, now remember, when it comes to X-linked recessive mutations, females can be affected, but it's a lot rarer for them to be affected because they have to have both of those X's having that mutant allele. In this pedigree chart, we don't see any females affected. They could, but it would just be rare. So it's okay that you don't see any females affected here because that could happen as well. But, it is possible for a female to be affected. However, how do you know this is an X-linked recessive trait that we're following? Well, because this is a dead giveaway. It skips a generation and then it pops up in the next generation. It doesn't have to skip a generation. It is possible that could pop up in the next generation, but it's oftentimes skips a generation. And you can see mostly the males are affected. Because we have an affected male, so this X has that mutant allele. Remember, the sperm parent will donate X's to daughters. So this daughter, one of the X's has a mutant allele. The other X comes from the egg donor. So the other X is going to have that wild type normal dominant allele. So this individual would be a carrier. Okay. So because it's a carrier, that means this egg donor now is going to give the X with the mutant allele to her sons. And the sons don't have an extra X to protect them, right? So the X, that's all they have is an X with the mutant allele. So the sons, they're going to be colorblind in this situation. Now, here's the thing. This mother has 2 X's. This mother could give the X with the mutant allele to her son, or she could give the X with the wild type dominant allele to her son. And that's why not all of her sons are colorblind, because it's 50-50. She could give either X chromosome to her sons. Okay, so that makes sense. Now, I do want you to be able to look at the 3 different Punnett squares we're going to go over. This is the first one, and be able to match them to the type of inheritance that we're following. And I also want you to know that the wording around these Punnett squares won't be there. So you'll simply be able to see how there's affected individuals. An X is a male, a typical male, and a circle is a typical female. And I just want you to be able to see by the pattern, be able to tell me what kind of trait we're following. And in this one, it's the X-linked recessive trait that we're following. Okay, any questions there? Okay, let's go on. We have the 2 more to look at. I believe they're all the way down here somewhere. Yes, let's start with not the mitochondria though. Let's go to the Y-linked genes. They're on the Y chromosome. And if there is any kind of mutation that happens in a gene on the Y chromosome, we will see a pattern where all the males of a sperm donor or producer, all of those male offsprings will have that affected gene as well. Because remember, the sperm donor is going to give the Y chromosome to their sons. So they don't have an option. Those sons are going to have that gene that has it. Now, obviously, daughters are not, they don't have Ys, they just have XX typically, right? So they're not going to be affected by any kind of a mutant allele that's on a Y chromosome because they don't have a Y chromosome. Okay, so, and you'll notice here it doesn't skip generations. It's the father is infected, has the condition, is affected by it because it's on the Y chromosome, and all of the sons also share that same situation where they have a Y that has the mutant allele. And then as they go on, you can see that it's goes on in the same pattern. All the sons have the condition. Now, obviously over here, why isn't that these individuals have the condition? Well, it's because this is a daughter. Daughters don't get the Y chromosome from their father, right? So this daughter doesn't have any type of Y mutation. So whenever this pair has offspring, none of the offspring are affected. Any questions there? All right. And then the last one we looked at was the inheritance of mitochondrial DNA. So mitochondria, remember all the mitochondria in our body, we inherit it from our mother. So whenever the gametes are being produced, remember how when the egg is being produced, there's 4 gametes that are produced, but 3 of them, they don't become mature to produce the egg. Instead, only 1 of the 4 become mature enough and matures to the point where it can be fertilized. And that egg is much larger than the other, or that cell is much larger than those other cells. And that's because it will have a larger amount of cytoplasm than the other cells that are produced. And part of what's in that cytoplasm is the mitochondria from, the egg producer, which is typically a female. So since all of our mitochondria come from our mother, and mitochondria, remember, their DNA is not in the nucleus. Their DNA is inside of the mitochondria. And so it's separate from what's happening and taking place in the nucleus. So if there is a condition that affects the DNA of the mitochondria, the mother will pass that on to all of her offspring. So that's why in this pedigree, we see how the mother's infected, or affected, I should say infected, affected. And we can see how all of those offspring also are affected with the same trait. Right? And over here, why don't we see any of these individuals affected by the trait? Well, that's because the father, even though the father has the affected mitochondria, fathers do not pass on their mitochondria to their offspring. Okay, so over here, this is an affected mother. So all of her children, all of the offspring will also show that same trait. They'll also be affected. Okay, so those are the 3 that I want you to understand and be able to identify. Any questions there? All right. Remember, I can't see the chat, so if you have anything you want to ask, just jump in anytime. All right. Let's go on to review the relationship between the crossing over event with recombinant chromosomes, well, chromatids or chromosomes, linked genes, and frequency of recombination. Now, it's because of the crossing over event that we get recombinant chromatids. I got to back up a little bit to find my slide on this, right? Because I think it's going to be up here somewhere. Yeah, I think there we are. We're getting to the point. So the crossing over event, here we see how it's taking place. This is where the crossing over event is taking place between 2 sister chromatids. Sorry, back up. These 2 here are sister chromatids, right? Because they are identical. However, once the crossover takes place, now they are no longer identical sister chromatids. The two chromatids that participate in the crossing over event are non-sister chromatids, right? Because this one here and this one here, they don't have to be identical because they can have different alleles. They do code for the same traits, but it is possible that they have different alleles. So they're non-sister chromatids. All right, so we end up with one of those chromatids is going to remain the same, and the other one is going to be a recombinant chromatid. Same over here. Okay, so it's the crossing over event that allows the shuffling of the alleles. So it helps with the diversity of the potential outcome of the gametes. Because of this, remember, we call genes that are on the same chromosome or chromatid, we call those linked genes. They can be inherited together into gametes, right? They could be passed on together into gametes, which would be the case for this chromatid right here. You could see it would remain the same configuration. However, it is possible for the original gametes to end up having something different. Right? So because of the crossover event, it allows for the chromosomes that are produced to potentially have genes that are from both parents on one chromosome. Okay. All right. And if these genes on one chromosome are further apart, it increases the chances that they're going to be separated due to the crossing over event. We can, as scientists, do experiments where we can look at traits that are controlled by genes, and we can look at the outcome as far as the percent of time that these traits are separated, appear separate or mixed in the offspring. The higher the percent or the frequency of recombination that we see gives us a clue that these genes, they're located further apart on the chromosome. Right, so higher frequency of recombination lets us know that these genes are probably located further apart on the chromosome. If they're close together, then it's less likely that the crossing over event is going to separate them. So we will see those traits inherited together more often. Right, so that would be a lower frequency of recombination number. Okay, let's see if there's anything, and then we use that information to make genetic maps. So I'm going to go, I know that's further down our list, but I'm going to go straight into that because we're in this spot of our PowerPoint right now. Let's see here. Let's go to a question. This is a good one. Let's go. I know we already did this in class, but don't look at the answer, okay? Let's go over and do it again from scratch just to make sure that you're comfortable with these types of questions. So we have some traits that are controlled by genes. You know, FV has to do with fused veins on the wings, okay? So we have like, I have a little index here to tell you what each letter represents as far as the trait that is controlled by the gene. Okay, so we scientists, they can look at the offspring and see how many times these traits are inherited together or are inherited separately. And so the frequency of recombination, remember, if there's a higher frequency of recombination, that indicates that those genes are most likely further apart on the chromosome. Okay, so there was an 11% chance, is what it's not really a chance, it's what the scientists observed, that 11% of the time, C and S, there was a higher chance of recombination taking place with these two genes. Okay, so because of that, we can go ahead and create a map that tells us where all the genes are. All right, so in making these maps, remember what we do is typically find the two genes that would be located furthest apart from each other. So that, since 11 is the largest number out of all of these, so that would mean C and S, I would put them on opposite ends of my gene. So first off, I just draw a line. Sorry, not of my gene, of my chromosome. So first of all, I just draw a line to represent my chromosome, and then I put gene C and S on opposite ends of that chromosome. And then I know that the rest of them are going to fall somewhere in the middle. Does it have to be that way? No, I could have had it so that like there could be like if this is my chromosome, it could be C is here and another one is slightly before it. But I'll just tell my students for this course, you just need a general understanding of how this works. And so I will tell you that when you identify the ones that are furthest apart on the chromosome, the other ones are going to be located somewhere in between them. Okay, just to make life easy for you. And then we can do that by simply, as soon as you put that C down, you can see, well, what else is close to C? Well, FV is very close to C. So I would just simply put FV somewhere close to C. And then I would look at S. What's close to S? Okay, well, what's this one here? So B is only 5.5 map units. Remember, we can call frequency of recombination. It's the same thing as map units. So I would just end up putting B somewhere, favoring towards the S side. And then once you have B in, you can just simply fill it in by looking at how close the genes are. All right, so go ahead and I'm not even going to show you the possible answer page, right? Because I have a page that says A, B, C, or D, and you can pick from that. I want everyone just take a moment, draw your chromosome. So just draw a line, put our C and S on it, and then try to figure out the order of the, basically you just have to figure out where the other two genes are located on that chromosome. All right, go ahead and take a moment to do that. And then I'll go to the answer page and we'll see if you got that right. I'm gonna go find my tea. I did make a tea this morning and I left it somewhere in my house. So I'll be back in like 30 seconds. All right, I found it. It wasn't too far away. All right, so let's take a peek at what the answer is. So those were our options. So your answer should have, you should have a line for that chromosome, and it most likely will say C, F, V, B, and then S. Do you have any questions on how I got that answer? All right, I think my next one shows it. Here it is. This is what my page would look like. Remember, you're always allowed to have paper during super quizzes. I just asked you to show me the front and back before you start, so I know that it's blank. Because I do have Punnett Square questions and questions like this in Super Quiz 7, I did make it so that you have 150 minutes to take Super Quiz 7, an insane amount of time, so you can just relax and just, have fun. I mean, can you ever have fun on an exam? I don't know, but you know what I mean. No stress, just keep calm. All right. So hopefully this isn't, the questions in the super quiz are not going to be any more complex than this. Okay, when it comes to this type of question. All right. Let's see where we're at. All right, using Punnett squares to calculate the probability of sex-linked trait in offspring. Most of my students are professionals at this by now because we've already mastered Punnett squares for the last super quiz. Let's go on and we'll practice one of our questions. I'm going to end up showing you the answers and I don't want to, so let's start here. Sorry. Let's see if I can find it this way. Here we go. Okay. Okay, this is a good one to practice. All right, let's see if we can all get the answer to this one. So we're looking at what proportion of males will most likely be white-eyed males offspring from the following fly pair. Now remember, the color of eyes for fruit flies, there's a gene on the X chromosome that controls that trait. So the mutant allele produces white eyes, where the normal wild type dominant allele produces red eyes. All right, so this is an X-linked recessive trait that we're following. The parents, the female is a carrier for the white-eyed recessive trait, and the male, it has red eyes. So the first thing I'm going to do is I'm going to make a Punnett square, and I'm going to put XX on one side and XY on the other, and I'm just going to fill in the inside. Okay, that's the first thing I do. So go ahead and do that. Make your Punnett square. Remember, anytime it has to do with sex chromosomes, first thing you're going to do, make a Punnett square, put the XX, XY, and then fill in the inside, right? And you should see when you fill in the inside that 2 of those 4 boxes are they're going to be female, right? And 2 of the other boxes, they're going to indicate probable males. So whenever you have XX and XY as individuals that are the gametes meet, there is a 1-to-1 ratio of the chances of having male or female children. All right, so hopefully everyone has their Punnett square already drawn up. Now you have to put the alleles on the chromosomes. Now, obviously, because this is an X-linked chromosome, you're not going to write anything on the Y chromosome because the Y chromosome doesn't have this gene on it. So on the X chromosomes on your Punnett square, you need to add the allele. The female, she is a carrier. So that means on one X, she's going to have a normal allele, a wild type allele. So on that X, I'm just going to do a capital letter N right next to it or above it. The other X for that female, though, since she's a carrier, I'm going to put a lowercase n to represent that mutant allele. Now, for the XY individual, it's a red-eyed male. So that X is only going to have the wild type allele. So on the male, I'm going to end up writing a capital letter N next to the X for the male. All right, and then you just fill in, all those alleles in your boxes, and it helps you to see what the probability is of having offspring that are carriers. It helps you and keep in mind, some questions, they ask specifically just to look at the males or just to look at the females. So remember, don't look at all the offspring and their probabilities of having the trait. Whenever the question asks you to look at only the males or only the females, you have to just concentrate on the males. And with this type of question, you're going to have 2 boxes, right, that have XYs in them. So you're only looking at those 2 boxes and seeing what proportion of the males have the condition. And I'll tell you right now, if there's only 3 options. The options, because there's only 2 boxes that produce males, right? So those 2 boxes, if they both have the trait, then it's going to be 100% of the males, or all of the males will have that trait. Or it could be 50% of the males will have that trait, or it'll be 100% of the males will have that trait. Those are the only 3 options when you're just looking at just the males from a Punnett square with this type of question. All right, so I'll give you a moment. Hopefully everyone filled in their whole entire Punnett square and they are able to see the answer, and we'll flip over to that. Where is this? All right, so the answer is half the males will be white-eyed, and this is why. Okay, so this is how my Punnett square would look, but just not with the fancy little cartoon drawings of the chromosomes, right? My Punnett square is going to say X, 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 Y. The red represents the dominant, no, we say, yeah, because affected is the white. So the red color, that would be where you have an uppercase N. So that's the wild type allele. And the female is a carrier, so that's why there's 1 red, one white. This would be uppercase N, lowercase n. And when we're just looking at the males, we see how this male is affected because its X has that mutant allele, where the unaffected male has that wild type allele. I really wish they made it's funny because the affected males, well, okay, yeah, they did it right. They made the gene colored white. So that helps us understand that affected males have white colored eyes. So that works for me. All right. Any questions about this? All right. We can do more of them if you want. I don't know if I have. This is a good one. Let's go ahead and do this one real quick. So we'll do one more. So green-red color blindness is an X-linked recessive trait in humans. If a carrier female mated with an affected male, what would the expected outcome be? Okay, so go ahead, take a moment. It's an X-linked recessive trait. So it's only going to affect those X chromosomes. Okay. All right, I'm going to take a peek at the answer now. Answer C, half of the daughters would be colorblind. Out of all those options it provided, half of the daughters would be colorblind. So you see how I solved it? I put my XX, XY, and then I put the alleles next to the X chromosomes. Remember, there's nothing next to Y because this is an X-linked trait. And then I filled in the inside. So in this box, I have a normal allele and a mutant allele. So we end up with a carrier female. However, this female has on both X's has that mutant allele. So this female is going to be an affected female. Now, when we're only looking at the daughters, right, we just ignore what's happening down here. We're only looking at the daughters, the females. It's half of the daughters would be colorblind. That's what we would predict from this pair. Okay, any questions there? All right. Why, and we kind of talked about this already, but why is it that XY individuals are more likely to be affected by X-linked recessive traits compared to individuals that are XX? Simply because individuals that are XX, both of their X chromosomes have to have that mutant allele, but because XY's individuals, they have a Y chromosome that is not capable of having that wild type allele on it, because that wild type allele, it only appears on the X chromosomes. Because of that, they are more likely to be affected by that X-linked trait. If that makes sense. Okay, so just keep in mind that it's this Y chromosome. It cannot, because it does not carry the traits that are on the X chromosome typically, right? So that affects its ability to have, it doesn't have a backup. That's how I like to think of it. XY individuals, they don't have a backup X like females do. All right. Okay, any questions about chapter 15 before I go on to 19? All right, let's hop on over to our viruses. And we start off with reviewing the basic structure of a virus. What do all viruses have? All viruses have a capsid, and this is a protein shell, a protein layer, if you want to call it. It surrounds the genome. So the genome is simply the nucleic acid, which is inside of that protein capsid. All viruses have this. Are they always this shape? No, because viruses come in all different shapes and sizes. So this is just a generic example, but all of them have a capsid, all of them have a genome. The genome can vary. It could be DNA or RNA, single-stranded, double-stranded, all that good stuff. But there's a genome within that capsid. Some viruses have an envelope, and remember where they get that from is they steal it from the cell they infect as they leave. This envelope, it can have viral proteins in it that help it with attachment with cells it will go on and infect later. However, the capsid as well too will have proteins that will also help it to recognize a receptor on this on their target cell, their host cell that they will infect as well. So that leads us to the third question, or the third option under chapter 19, which says a review what determines the host range of a virus. So not all viruses can affect all cell types. What makes the difference is the proteins that are on the virus as well as the proteins that are on the host cell. They must interact like a lock and key, how lock and keys do, right? They must fit together, kind of like puzzle pieces. If they interact, then that allows the virus, you can kind of think of it as it gives permission for the viruses to come into the cell. And on the host cell, we call that protein a receptor. On the virus, oftentimes we call these, sometimes they're spike proteins that interact with the receptors. And, but to keep it simple, if the receptor that interacts with that protein is there, then that virus can affect that cell type. This is why with COVID-19, some cells cannot be infected by COVID-19 simply because those cells don't have the correct receptor. And for COVID-19, that receptor is ACE2. And that is seen typically on those cells, that the epithelial cells that line our respiratory tract, right? In our nose, in our airway, and deep down in our lungs. All right, so what makes that big difference when it comes to the host range, the type of cells, and the species that the virus can infect has to do everything with those proteins on the surface of the virus and the surface of the host cell. Okay. I do have a question where I want you to be able to recognize how these viruses come in different shapes and sizes, and just to recognize the different types of viruses that are out there in particular. So this page is going to be very important to study because you have an example here of viruses that are enveloped, and the example that they give you is a coronavirus. But influenza, herpes, and HIV, they also are an enveloped virus. So I put a little note down there so you can understand that. This example here, it is of a non-enveloped virus, and I give a little example. HPV falls in that category as a non-enveloped virus. Tobacco mosaic virus is a virus that only infects plants, and it has a very unique shape and structure where the capsid, it actually is intertwined with the genome. So it's actually in between the layers of the capsid. So it's kind of cool. Very unique. But the coolest one of all, this guy here, right? The T4 bacteriophage is very impressive. It is the most complex out of all of the shapes. So when you see this structure, just know this virus, it can only infect bacterial cells. It's not, this T4 bacteriophage is not going to affect cells in our body, but it can infect bacteria. It's one of the reasons why populations are controlled in the environment, because anything that is cellular most likely can be affected by viruses. So viruses, even though we associate them with harmful effects, for us, right? But we can also look at them as being helpful to help control some of those pesky things in our environment that cause problems and keeps them under control. Right, so just be able to identify, I would know the name of the example virus that I provided, as well as what are we looking at here? We're looking at a virus that only contains a capsid and a genome. There's no envelope. So it's a non-enveloped virus versus one that has an envelope. Okay, this type of virus would typically infect a plant, tobacco mosaic virus, and this one here would typically infect a bacteria cell. Okay. All right. Next thing we have review the characteristics that the Baltimore system of virus classification uses. Right here. Okay. One of those most useful classification is the Baltimore system, which is based on two characteristics. The type of nucleic acid the virus contains, double-stranded RNA, single-stranded, double-stranded, positive sense, negative sense. We're going to go over that too. Also, how the messenger RNA, which produces those viral proteins, is synthesized. That is regarding the positive sense and negative sense, particularly. Okay, because when we look at how it's laid out, we notice that anytime there's a single strand of nucleic acid, all these, right? We notice that there's either a positive or a negative sign associated with it. When it's double-stranded, there's no positive or negative sign associated with it, right? It's only when there's a single strand, and that's to help us identify if the nucleic sequence that's in that single strand, if it matches, if it's the same sequence in the mRNA strand that is used through translation to make viral proteins. If it's the same sequence, then we call it a positive sense genome. Now, keep in mind, this is DNA, this is RNA. So there is going to be a slight difference, right? Because the DNA is going to have thymine, the RNA is going to have uracil. Okay, yeah, I understand. So it's not identical, right? But if you looked at the order of the nucleotides, if this said A, G, down here it's going to say A, G, right? It's going to be the same sequence, except for that little caveat with the thymine and uracil. Okay, so we know how the characteristics that the Baltimore system uses, and then there is down the space. Do you understand? I don't see it written down here, but I thought I did write it down here. Do you make sure you understand what is meant by negative sense and positive sense when it comes to the Baltimore system of virus classification? Okay. And if you want, oh, hello. This is the best one to study when you're trying to understand what positive sense is. If you still need further clarification, right? So the viral nucleic acid, so the genome that's contained within the capsid, it's positive sense if that sequence matches the mRNA sequence that will be used to make viral proteins. But remember, the viral genome, it could be RNA or DNA. So as long as it's a single strand, it is possible for it to be positive sense RNA or positive sense DNA. Obviously, the DNA, if it is DNA, that can't be directly used in order to make viral proteins. The positive sense DNA strand, it has to first be converted through translation or through a certain process. Transcription. Gotta keep my words straight, in order to become mRNA, where the positive single strands RNA genomes, once they enter the cells, they're ready to go. Like they can immediately interact with ribosomes and translation, can immediately begin to make viral proteins. So positive single-stranded RNA viruses, they are able to, I don't want to say do a lot of damage, but they're able to affect the cells a lot quicker than ones that are infected with other genomes that have to go through a process to make or synthesize the mRNA strand. Okay, so that's positive sense. So whenever, as long as that sequence that's in the genome matches the sequence that's in the mRNA strand, then we call it positive sense. And then if that strand of nucleic acid in the genome is complementary to the mRNA sequence, then we call it negative sense. If it's complementary, then that strand can be used in order to transcribe the mRNA. So we can call it a positive sense mRNA is used to make more viral proteins. So if we look here, we already went over this, right? If it's, so this one here, group 5, influenza, it's a negative sense single-stranded RNA. Why is it negative sense? Because the nucleotide sequence does not match that of the mRNA strand. It's complementary, so it can be used in order to produce the mRNA strand, but it's not the same sequence. So it's negative sense. Okay. All right. Review the differences between the lytic pathway and the lysogenic pathway of a bacteriophage. Down here. I passed it, didn't I? Diversity, classification, and then we do replication right here somewhere. Let's go to this page. Whoop. Okay, so this is specifically with bacteriophages, right? This is how that T4 bacteriophage can infect a bacterial cell. There is a similar process when it comes to viruses that can infect eukaryotic cells. They're similar. Obviously, there are going to be differences as well, but it's very similar. So let's focus on this one here. So we see how that phage, that bacterial phage, that little sign there is lambda. We see how it can inject the DNA into the bacterial cell. Now, when it comes with viruses that infect eukaryotic cells, they don't inject their DNA in. Instead, that whole entire virus, if it can interact with that receptor on the cell, it will trigger endocytosis or plasma membrane fusion, which will allow that virus in. So in this case, though, we see how that genome is injected within that bacterial cell. And then two things can take place. Either it can end up going the lytic pathway or the lysogenic pathway. In the lytic pathway, that's when that genome is immediately going to be replicated, and it's going to go through the process of transcription, translation to produce lots of viral proteins. And so you have lots of viral genomes, lots of viral proteins, and then they're going to self-assemble spontaneously. It's kind of like if you had a handful of magnets in your hands and you were to jiggle them around, eventually all those magnets are going to self-assemble according to, negative and positive regions. And eventually, when a lot of those virions are produced, they're going to burst the cell and be released, and then they can go on and infect neighboring cells. The other possible pathway is a lysogenic pathway, where that genome is going to be incorporated into the genome of the host cell. And you can kind of think of this as the genome is hiding out and it's kind of behaving itself, it's not active, it's dormant. But by being dormant like this, every time this bacterial cell goes through binary fission and creates daughter cells, all of the progeny produced by this bacterial cell is there. The genome is also going to have a copy of the viral genome in it as well. So this ends up in a situation where there's lots of bacterial cells that has the viral genome in it. And then through some kind of stressor in the environment, it is possible for that viral genome to then be triggered to go to the lytic pathway. And then it'll end up, with a lot of virions being released into the environment to infect a lot of other cells. Okay. With our cells, we don't say that the viruses take either a lytic or lysogenic pathway. We say it can take a lytic-like pathway or a lysogenic-like pathway that is similar, where it is either dormant within the cell or active within the cell. Okay, that's the only real big difference. This page here is important because it just highlights those little differences whenever a virus infects a eukaryotic cell. Remember, the big one is that once that virus attaches, it enters the cell through endocytosis or membrane fusion. Once inside of the cell, uncoating takes place. Uncoating is where that the capsid is disintegrated, disrupted in some way to allow the genome to escape the capsid and to be released into that cell. Then host enzymes replicate the viral genome, put a little period there. And also enzymes transcribe the viral genome into mRNA. The mRNA is used to make viral proteins. The virus self-assembles and then is released. And it can be released by lysis, which is where the cell bursts, or it's possible for the cell not to burst, not to be killed. And the viruses, the virions that are produced, can be slowly released from the cell. So it doesn't always have to cause that cell to immediately burst and be killed, but it can allow the cell to survive for longer and just slowly release those virions by budding off. Okay. There is this video right here. This video at this website will really help you understand that process of how those viruses affect eukaryotic cells with attachment and the whole process inside and how they can be released. Okay, so I would spend some time. It's not long. It's just a cute little short video. Help you understand all that. All right, let me see what else we have on this. Review the structures and genomes of the following virus types: COVID-19, influenza, HIV. So we have a little bit of information about them on that one page that shows you the different shapes and sizes. Right up here. Right here. Okay, so remember, I do give some information here on what they are. We know that COVID-19 is a type of coronavirus, and also influenza, herpes, HIV, they all are enveloped. So I would, when you're writing down your characteristics and structures of these viruses, I would simply make sure you do note that COVID-19, influenza, herpes virus, HIV, they are enveloped viruses. All right, what else are they? For the rest of that information, I would hop down over to the Baltimore System of Virus Classification, because we can see how we have coronavirus, influenza, HIV. They're all there. So besides having an envelope, right, we have coronavirus, which COVID is a coronavirus, is positive sense single-stranded RNA. Okay, so I want you to be able to know the type of genome it has. Flu, influenza, has an envelope and is negative sense single-stranded RNA. HIV, it's a retrovirus. What are retroviruses? Retroviruses use reverse transcriptase, that's an enzyme, in order to cause their genome to go from RNA to DNA. So that's backwards, right? That's why it's called retro. That's backwards from this central dogma of molecular biology. So that's why it's called a retrovirus, because it'll have some process where it will go from mRNA to synthesizing DNA, right? Notice that group 7, it starts off as incomplete double-stranded DNA, but eventually during the process of eventually creating that mRNA strand, it goes through a stage where it does have RNA that is used to produce DNA. So it's also a retrovirus. Right, but we have all the genomes up here. for our, coronavirus, aka COVID-19, influenza, and HIV here. So I would just make sure you're very well aware of their genomes. Okay. Mutation rates. What does that mean for us? And how does that impact the need for vaccine boosters? Well, some of those viruses, their genomes mutate very quickly. It makes it very hard for scientists to develop a vaccine, because as soon as they develop a vaccine that allows the immune system to have an immune response that recognizes the specific proteins that are on that virus, those proteins can change shape. Why do they change shape? Because the genome of the virus mutates. Anytime you have a change in the nucleotide sequence, which is a mutation, right? Or caused by mutation, then ultimately it can affect how the protein folds and the shape of the protein. All right, so viruses mutate quickly because many of them lack proofreading enzymes. Our bacteria, so on our chart here, we see bacteria. Anything that's higher up on this chart, because this is mutation rate, so higher up means a faster rate of mutation. E. coli, a bacterium, is very low in comparison to our viruses. They're a lot higher. Okay. So it's because of the mutation rates much higher in viruses. That's why it's tricky for us to make that like vaccine that works for a long time, right? Even in the middle of the flu season, it's possible for the flu virus to mutate, which, makes it very tricky. Now, let's see here. I have a page, it's at the very bottom on here. Antigenic drift. So that's what we call it whenever the genome within the virus mutates and produces those genomes that produce a protein that has a slightly different shape. Okay, so think antigenic drift whenever you're thinking about those small changes that take place to the genome, producing those different shape proteins, which allow this virus now that has mutated can escape our immune system, because our memory B cells, they no longer recognize the antigen. When we're talking about antigens, remember, we're talking about something on the surface of the virus that can interact with antibodies. So antigens are going to interact with antibodies, right? But it's not going to recognize that. It's not going to fit lock and key anymore. So the immune system, it has to basically start over with recognizing it again and going through the proper process of producing a new army of memory B cells. And that takes time. So that's why whenever it mutates, we can end up getting the flu again. All right, antigenic shift, that has to do with two strains mixing. So we have a strain here that affects birds, avian strain, the bird flu. An individual, a human, can be infected with the human version of the flu, and maybe they're working around birds, so that their system ends up having the bird flu. It ends up being infected with that bird flu. Typically, this bird flu is not going to be able to get into our cells, right? Because these proteins, they need a specific receptor that are not found on our cells. So this genome can't get into our cells. It can't affect our cells. However, if through the mixing of the two genomes, it's able to get that genome inside of the virus that affects humans, right? Which can happen if the individual already has the human strain and then is infected with the avian strain. Then you see how that genome gets into the capsid, and now you can see how this virus can affect human cells, because it does contain, in this example, those pink colored pinky purple colored proteins are able to interact with the receptor on the human cell. And now you can see how, yes, there's green and there's pinky purple, but as long as it can interact with the receptors, and it can because it has that pink colored protein, it's going to allow this genome to now get into the cell. And this is going to cause a new novel strain to be formed. And in these types of situations, because it's new, it's brand new, it's not circling out in the population, nobody in the population has any type of immunity to this. So you're going to see things like pandemics happen when antigenic shift takes place. All right. Okay, I have a little bit on CRISPR and a little bit on vaccine and immune response to go over. Let's leave CRISPR for last. Let's go on to vaccines. And it says, how do vaccines protect individuals from the disease? Look at the important slides pertain to what I need you to know. They're right here. I think it's actually above this. How vaccines work. That's the mRNA. All right, in general, how do vaccines work? They train your immune system to recognize and fight pathogens before you ever encounter the real infection. All right, so how do they work is that they usually contain some kind of harmless form of the virus or bacterium, or they can contain pieces of the virus or bacterium. And those harmless forms or those pieces, they can't cause you to get the disease. But they do allow the antibodies that are already circling in your system. Remember, we have like how many antibodies we have circling in there? It's in the billions. Just want to make sure I don't say the wrong number. 10. So we have an estimated 10 billion different antibodies that are already circling in our bodies. They're already there. So this immune response is what protects us from the second time our body is introduced to that virus, this side of our diagram. However, in our body right now, you have a bunch of, we call them naive B cells. They are already circulating in your body. There's estimated 10 billion different antibodies that are already in your body. But they're not, these naive B cells, they aren't the ones that fight the infection. They are there simply to identify that something's there that should not be there. There's an invader in the body. So once they come in contact with an antigen that is a part of a virus or a bacterium, they're able to bind to it. So in this example here, it just so happens that this B cell with this antibody type has a region that can bind with this antigen, this intruder. So once it's recognized, then it's going to end up going through cell differentiation, where the B cell is going to differentiate into plasma cells, which those plasma cells are going to produce a lot of these antibodies. They're going to be secreted so they can surround the virus, blocking the ability of that virus to interact with the receptors on the host cell. What's also going to take place is some of those B cells are going to differentiate into memory B cells. These memory B cells are going to stay in your system for years, and there's going to be a whole army of them produced, which means they're constantly going to be on the lookout for that same invader. And as soon as the second exposure takes place, right away, those antibodies are going to be able to recognize it because you have a lot of these circulating your system now. So there's a good chance it's going to interact with it very shortly after you're infected. And immediately, those memory B cells, once it identifies that this invader's back, then cell differentiation is going to happen again. And a lot of those plasma cells are going to be developed, secreting a whole bunch of those antibodies to interact with the virus, not only blocking it so it can't get into our cells, but also flagging it so that our other immune cells can come and destroy the virus. Okay, so that's how vaccines work, is simply introducing to our immune system what the virus or the bacterium looks like, right? Because if it interacts with that antibody, then it's going to produce this type of response. It's going to cause an army of memory B cells to be produced. And then, therefore, if the real pathogen then is introduced into an individual, it's going to be recognized right away. And sometimes you don't even know that you've been infected. The immune system will take care of it right away with that second exposure. Sometimes, though, you might feel a little off for a couple days, and that's just because your immune system is working a little overtime, right? To go through cell differentiation. Lots of those antibodies are being secreted. So in your lymph nodes, they're going to feel swollen because this type of actions taking place. So you could feel rough for just a day or two, but then it's quickly taken care of. So the first time you get sick, or well, it's a bit different. Like if you get sick and it's the first time your body's ever been exposed to that virus and you've never had the vaccine for it, right? That's why you feel rotten for a week or two, because it takes time for your body to go through the process of the cells identifying the antigen, and then through cell differentiation. This whole process takes time. So that's why it's a lot longer of a process when you get sick the first time. Second time, though, it's quite fast as far as how long it takes for those antibodies to be produced. Okay. All right. On to CRISPR. What do you need to know? Okay, let's talk about here. What do you need to know about CRISPR? Well, the history about how we discovered it is simply some bacteria and archaea species. Scientists realized that there were these little segments of nucleic acids within their genomes. They were about like 20 to 50 base pairs long. They're just tiny little sequences that were in there and sometimes repeated. So they were just, scientists were curious about these sequences. And that's how we came up with the term CRISPR, because those little tiny sequences, let's see if I have my page here. They stand for clustered, regularly interspaced, short, palindromic repeats. Okay, that's how we get the term CRISPR. It has to do with those little sequences that we see within those genomes of viruses and bacteria. Or not, sorry, not viruses. Within bacteria or archaea. Sorry about that. And here we go. So since we know that bacteria, the reason why they have these short sequences is because whenever they are first infected with a virus, they have a process of actually taking a copy of, they take a snippet of the viral genome, just a little portion of it, and they embed it into their genome. Keep in mind, they don't put in the whole entire viral genome into their genome. That would cause a lot of damage, right? Because then all of a sudden, every time that cell goes through division, it would copy that viral DNA. So that's not what happens. With CRISPR Cas9 system, what's taking place is that it's just a tiny short bit or section of that viral genome is then integrated into the genome of that bacterial cell. And by doing so, it has the ability to recognize the invader genome again in the future. Okay, so it works by also within that bacterial cell is a gene that codes for the protein Cas9. Cas9 interacts with RNA that is produced from the viral genome snippet that's inside of that DNA now. So what scientists do is they can actually make a DNA plasmid. Plasmid is simply just a small piece of nucleic acid sequence that contains some genes. Keep in mind, plasmids, they can be just normally found within bacteria cells. However, scientists can also genetically engineer plasmids. Okay, so in this case, a scientist will engineer a plasmid with a DNA sequence that codes for the guide RNA and for Cas9. So this is what the scientists inject into the cell. So in the cell, there's going to be a gene that the scientists want to edit. So they inject it with this plasmid. This plasmid, through the process of transcription, is going to produce this piece of RNA, which has a complementary region to the target DNA, the one that they want to cut. Right? On this plasmid also encodes information that can be used through transcription and translation to produce Cas9. So the guide RNA and Cas9, they associate together, and then they can recognize the target DNA, and then Cas9 will cleave the target DNA. So now there's a gap that is produced in this genome. Then it's important at this time, scientists will then introduce what's called template DNA, editing template DNA. This editing template DNA is complementary to the sequence that scientists want to have within the gene that they're trying to edit. So this complementary DNA strands, they are used in order to fill in that gap with the correct sequence or the sequence that they would like to see in that genome. Okay. And it results with a edited DNA strand with an altered sequence. Okay, so when you're studying the different processes of this, I do have information about each one. I have a little bit of information, spread out over several slides, pretty much what I just said. So if you on your own time want to go over these pages to just kind of think through the process, this is an excellent way to study it. All right, so just kind of takes you through it slowly. Now, I do have 2 videos in D2L that also talk about the process and go through it. They're excellent. So that'll also help you get a deeper understanding for that process. All right, so let me see if there's anything else I want to ask. Any questions before I review my notes? All right. All right, let me look over my notes and see if there's any additional information I want to provide. Give me one more moment. Okay, I want to talk about HIV just a little more. Let me see if there's anything else. Okay, cool. All right, so let's go talk about HIV just a little more. Oh, I see that one. Oh, these are my extra notes that you don't have. These are the ones I didn't incorporate into our lecture this semester. Do the old fashioned way. Here, this is HPV. Did I have HIV? Oops. Here we go. This is the one I want. Okay. So we already talked about how HIV uses the enzyme reverse transcriptase to make a DNA copy starting with an RNA genome. So that's important. But how does it get into the cell? In order for the virus to enter the cell, it needs to interact with two receptors. Remember, HIV infects T cells, which T cells are very important in our immune system because they help, you can think of them as they help kickstart the whole immune response. So without T cells, the whole immune system suffers. So HIV specifically infects our T helper cells. And in order for it to gain entry into the cell has to have these two receptors. One receptor is a CD4 receptor, the other receptor is a CCR5 receptor. So it's called a co-receptor because the two are needed. And interestingly, though, some individuals have a mutation in the gene that codes for the protein that produces that protein that's involved in that receptor, which is CCR5. So in individuals that have this mutation, their CCR5 protein, that receptor, is slightly different shaped. So that makes it so that it cannot interact with the proteins that are on the surface of the virus. So this allele, this mutation, it's caused by the allele which we refer to as delta 32. We call it delta 32 because when we looked at the mutation, there's actually 32 base pairs that are deleted in that coding sequence, in that gene that's responsible for synthesizing or producing the CCR5 receptor. So because this receptor has a different shape, it results in the inability of surface glycoproteins. So those proteins, if it says glycoproteins, it's simply it has some carbohydrates attached to the protein. So that's why we call it glycoproteins. But it prevents HIV from binding to the receptor, that CCR5 receptor, on the host cell. So that makes it very difficult for the HIV to infect those target cells if the individuals have that mutation, which we call delta 32 allele. All right. This is really interesting because that's why we find that some people are simply immune to HIV because the virus has a very hard time entering those individual cells. So scientists, this was very interesting when they were able to discover this. Its prevalence throughout the world varies, and it's approximately 10% in European populations. That's the highest frequency we see it. So it just varies throughout different populations throughout the world. But this is the reason why some individuals are naturally immune to HIV. It also explains why certain individuals, it's a similar process where when you look at any type of viral disease, why are there some people that aren't affected by that disease? They're just naturally immune. Well, because it has to do primarily with the receptors that are on their cells. They are slightly mutated to a point where that virus cannot gain entry into their cells like it can for most of the other people in the population. Okay. All right. Any questions about the allele delta 32 and how that inhibits the ability of HIV to get into the cells? Okay, good. So hopefully most of this is just, it's all making sense. I feel like this topic, both chapter 15, chapter 19, and CRISPR and the immune response are usually my students find that they have a little easier time understanding this content. So as long as you go over what we talked about, and if you practice some of those questions, watch some of those videos, you should be fine. Okay. I will be around. I will be checking my email often, especially because I want to make sure everyone's grade looks right. I will send out announcements when I finalize all the grades in D2L, and I will let you know because I at that time I'm going to ask you to check your grade. And I'm going to give everybody a day or two in order to check their grade so that if anything looks weird or out of place, you just let me know. Even after once I submit the grades to AliExpress, to KSU, and they put it in AliExpress, if there is anything wrong with your grade, I do have a process where I can fix that. Okay. It takes like a week, sometimes or two, but I have to submit a form in order to officially change your grades once I put them into AliExpress. In 5 years of teaching, I only had to do that one time. So I'm very careful when I do that. The one time it did happen was simply because Excel rounded the grades differently than what D2L did. So it caused someone's grade to be different than what it said in D2L. So I just want you to know that never panic when it comes to the end of the semester grades. If you see anything wrong, send me an email and I promise I will fix it as quick as I can. All right. Is there anything else anyone wants to ask or mention before I sign off?
Professor,
Yes.
so after this, will you still have like you know how the office hours things, like if we have questions,
Yes.
do you think will you still have that or no?
No, so the test closes on Thursday, and Thursday I usually have office hours, but I won't have any of my office hours after that. But what I will still have is I will make it available for people to, well, I shouldn't say that because I don't know how close I'm going to be monitoring it, because you can always book an appointment using that link that I provide
Yeah,
in D2L.
yeah.
I think honestly, because you know, I won't be as closely monitoring my email once school is over, once class is done. The best way to get in touch with me is to just send me an email. I will, like, every once in a while check it, and then we'll set up a time to meet. Um, yeah, I will, especially, and I don't know. So next week and the week after that, I still will very closely be monitoring my email. But then once we get to like the week of Christmas and whatnot, I usually like to take a little vacation time, so I'm not checking it as closely. So just feel free to email me if you have any problems over the next couple weeks. For sure, I'll get that email and we can set up a time to meet. That's probably going to be the best way.
Okay,
All right.
thank you.
Yeah, no problem. Good question. Anyone else? All right. Well, then I'm going to sign off and say, it's been fun and I've really appreciated having you all students this semester. Thanks for making a great semester, and I hope you all have a wonderful break. All right. Take care.
So thank you.
Very welcome. Bye. Oh, wait. Why am I doing that? Okay. Sorry. Um, all right. Here we go. All right. Take care. Bye.